Hello everyone, hope things going well. Today I will give a presentation with my classmates Li Liangkang and Mamu. Our topic is to discuss how to enable high throughput data for diseases diagnosis. Firstly, please allow me to give some brief introduction. What is high throughput data? Generally, it involves rapidly processing and analyzing vast amounts of biological information, typically through automated and advanced computational methods. Here are two examples in genomics and in drug discovery. Also, these techniques are super important in diagnosing diseases. They are they have pushed us towards personalized medicine, where treatment and diagnostic are more based on individual genes. Based on this, we have to focus on high throughput technology with advanced computational tools and bioinformatics. Then let's look back at some history. In 1965, Robert Halley sequencing the first tRNA, laying the foundation for molecular genetics. In 1977, Frederick Sell developed cell sequencing, introducing the dideoxy chain termination method for DNA sequencing. And in 1987, automation of cell sequencing incorporating fluorescent dye labeling and computer-based data analysis. Then in 1996, Pyro sequencing, marking the beginning of modern high-throughput sequencing. In early 2000s, various next-generation sequencing platform developed, and recently in 2012, Oxyfood nanopore techniques introduced systems like grad iron and minium iron, utilizing the biological.
The slide is high throughput data for disease diagnosis. So I taking for the one example which is the inside the medical. So the first point is medical genome sequencing. So genomic sequencing having enormous impact in the medical fields. So it's very important for the medical medicine field. Until now, the cost and the throughput limitation made general clinical application infeasible. So the high throughput sequencing has helped a lot of diagnosis and very highly genetically heterogeneous disorder, which is the X-linked intellectual disable congenital disorder of the glycosylation, and detect the carrier status for the rare genetic disorder. So the first example we can see is genome sequencing in cancer. So the cancer is a genetic disease, which is the person which is in the predisposition, and also can the cancer also can grow inside the somatic body. So the high throughput sequencing can understand the complete disease or the cancer in the genetic. So the high throughput sequencing or data can know about the somatic alerts alternation such as mutation, amplification, deletion, and translocation. So I give the example of the disease is about the breast cancer. So the researcher they found it not only for the for the breast cancer for the like BRCA1 or 2, they found it in the another gene which is the RPS6KB1 and VMP1. They will fusion the gene. They also can occur breast cancer with prognosis. So the researcher also using the pair on sequencing and on cancer, he can observe the not cancer human. The result will be inside the human body. They will inside the gene. They will find it in inversion, deletion, insection. Then the other which the human which is have having cancer, there's inside they can observe the gene inside the cancer human, which is duplication, translocation, and some of the complete rearrangement of the gene. So next is genome sequencing also can find some rare disease or mystery disease. So so the researcher also uh, need some high throughput analysis to prove the successful in the diagnosis of the rare cancer in the world, the whole genome and the exome sequencing. So this sequencing also can selecting the optimal individual treatment option for patient. So I give the one example for the disease which is the X-linked lymphoproliferative disease, which is the XLP. Is caused is the person that caused by this disease is Nicholas Walker. Is a famous patient. So he's the first one alive because of the DNA sequencing. So the Nicholas Walker they received a bone marrow transplant after his genome sequencing get mutation on the X chromosome that lead him get some inherited immune disorder. So after that, they take a treatment. So the vocal successful has drip, and his inflammatory bowel disease also very reduce it. So with the multiple example adding diagnosis and treatment of true medical cases, high throughput sequencing in medical care is useful but still having limitation. So because of the gene barrier and the effect of the human health. So next is the personal genome sequencing can detect medication actionable risk, means can note early for the diagnosis. So the high throughput technology they will focus on specific omic network, example like DNA, RNA, protein, and metabolites. So the researcher already update the high throughput technology, which is the multi-omic. Not they can focus on one, which is the example like they are focused on DNA, RNA, and protein metabolism. Observe they can observe like DNA, RNA, protein in the same time. So they can also can identify the unknown disease with complex sequencing. Sorry. So now the medical staff using this multiple omic data set to get the higher accuracy of the result. So this one, I give the example. The genome sequencing also can track the emergency type 2 diabetes. So we know the type 2 diabetes of the because of human behavior. So and then some if they were a long time, if long time they were the how to say the insulin will be dropped. Okay, so we how to see the gene sequencing. They see they got two gene, which is a TCF 7L2. They will affect the insulin process and the glucose produced. Another one gene is a ABCC8 gene, will help to regulate regulation of the insulin gene.
Hi everyone, I am Mohammad Al Mamun. Now I am presenting the uses of the uh, high throughput data in disease diagnosis. The advantages in the disease diagnosis, the uses of high throughput data in various sectors, like it includes molecular profiling. A more thorough knowledge of disease is possible because high throughput methods which provide a full perspective of the molecular landscape comprising proteomics, metabolomics, transcriptomics, and genomes. And it has a great access to finding the biomarkers. The discovery of certain molecular biomarkers linked to disease that is made possible by high throughput data. These biomarkers may be used for prognosis, early identification, and customized treatment plans. And we can use the high throughput data in also personalize the healthcare, the ability to customize healthcare to each patient's unique traits thanks to high throughput data results in treatments that are more accurate and successful. And it has a great implementation in the development of the new drugs and drug discovery. Now we are in the point of disadvantages of high throughput data in disease diagnosis. The first point is the multiplicity of data and its interpretation. When it comes to accurate interpretation, high throughput data is often difficult to understand and calls for experienced bioinformatics professional. There are several challenges that might arise when attempting to analyze and extract relevant information from huge databases. And also there is also another disadvantages, like it's like the cost, the fees, and available resources. When it comes to implementation and maintenance, high throughput technology may be rather expensive. There is the potential for a limiting element to be the expenses that are related with the equipment, reagents, and computing the infrastructure. And also it has some disadvantages in quality, in maintaining the quality of the data and its there and the standardization. It may be challenging to guarantee the quality and standardization of high throughput data across a variety of platforms and labs using the same methodology. Results that are not dependable may be the consequence of inconsistent data quality. And also there are some disadvantages, the data validations. Thank you.
To summarize our topic, the cost effectiveness of high throughput technologies make large-scale screening and personalized medicine more accessible, which is good news for human health. But there are still challenges like the massive volume of data generated require complex bioinformatics pipelines and specialized skills for interpretation. So the future of high-throughput data needs the integration of multi-omics data and long-time monitoring with new equipment and IT. So that's all. Thank you so much for your attention.